amino acids, with each amino acid being linked to another via a peptide bond. Peptide bond is formed when the carboxyl group of one amino acid links to the amino group of another amino acid with the loss of a water molecule. N-terminal, end of protein structure with a free amino group. C-terminal, end of protein structure with a free carboxyl group. Nitrogen content. Proteins consist of 16% nitrogen, which differentiates proteins from carbohydrates and lipids. Primary structure. The amino acids are linked to each other through covalent peptide bonding in a specific sequence to form a polypeptide chain. Secondary structure. The polypeptide chain winds to form alpha helixes and beta sheets through the formation of hydrogen bonds between CO and NH groups of the peptide bonds. Tertiary structure. The coiled polypeptide chain folds upon itself to form a three-dimensional structure through the interactions of the R groups of the amino acids. Such interactions include the formation of disulfide linkages, hydrogen bonds, hydrophobic interactions, and van der Waals forces. Quaternary structure. Two or more folded polypeptide chains bind to each other through hydrogen bonds and electrostatic interactions to form a functional protein. Simple proteins. Polypeptides composed of only amino acids. Globular proteins, symmetrical, compactly folded polypeptide chains, for example, albumin. Fibrous proteins, elongated, asymmetrical polypeptide chains, for example, troponin and collagen. Metalloproteins, protein with a metal prosthetic group. Conjugated proteins, composed of protein and non-protein components. Prosthetic groups are commonly metal, lipid, and carbohydrate in nature, for example, ceruloplasmin. Lipoproteins, protein with a lipid prosthetic group, for example, cholesterol, triglyceride. Glycoproteins, protein with 10 to 40% carbohydrates attached, for example, haptoglobin. Mucoproteins, protein with more than 40% carbohydrates attached, for example, mucin. Nucleoproteins, protein with DNA or RNA nucleic acids attached. Energy production. Proteins can be broken down into amino acids that can be used in the citric acid cycle to produce energy. Water distribution. Maintain the colloidal osmotic pressure between different body compartments. Buffer. The ionizable R groups of the individual amino acids of a protein provide buffering capacity by binding or releasing H ions as needed. Transporter. Binding of proteins to hormones, free hemoglobin, lipids, drugs, calcium, unconjugated bilirubin, and so on, allows movement of these and other molecules in the circulation. Antibodies. Proteins that protect the body against foreign invaders. Cellular proteins. Function as receptors for hormones so that the hormonal message can activate cellular components. Some hormones are protein in nature, for example, adrenocorticotropic hormone, follicle-stimulating hormone, luteinizing hormone, thyroid-stimulating hormone. Structural proteins. Collagen is the fibrous component that maintains the structure of body parts such as skin, bone, cartilage, and blood vessels. Enzymes, catalysts that accelerate chemical reactions. The liver synthesizes most of the plasma proteins. Plasma cells synthesize the immunoglobulins. Proteins are synthesized from amino acids, with one amino acid linked to another through the formation of a peptide bond. When proteins degrade, their constituent amino acids undergo deamination with the formation of ammonia, which is converted to urea for excretion in the urine. Some cytokines released at the site of injury or inflammation cause the liver to increase synthesis of the acute phase reactant proteins. This is a nonspecific response to inflammation that may be caused by autoimmune disorders or infections, as well as a nonspecific response to tissue injury from tumors, myocardial infarctions, trauma, or surgical procedures. On the other hand, some proteins will decrease in concentration and are referred to as negative acute phase proteins, including prealbumin, albumin, and transferrin. Immunoglobulins are humoral antibodies produced in response to foreign antigens for the purpose of destroying them. Reference ranges: total protein 6.5 to 8.3 g/dl, albumin 3.5 to 5.0 g/dl. In general, changes in total protein concentration are associated with hypoproteinemia caused by urinary loss, gastrointestinal tract inflammation, liver disorders, malnutrition, inherited immunodeficiency disorders, and extensive burns. Hyperproteinemia caused by dehydration, increased protein production associated with monoclonal and polyclonal gammopathies, and chronic inflammatory diseases associated with paraprotein production. Prealbumin, also termed transthyretin, is an indicator of nutritional status and is one of the proteins that transports thyroid hormones. Decreased in liver disorders, inflammation, malignancy, and poor nutrition. Increased in steroid therapy, chronic renal failure, and alcoholism. Albumin is synthesized in the liver and has the highest concentration of all plasma proteins. Albumin binds many analytes for transport in blood, including unconjugated bilirubin, steroids, ions such as calcium and magnesium, fatty acids, and drugs, and it significantly contributes to plasma osmotic pressure. Decreased in liver disorders because of decreased production, gastrointestinal disease associated with malabsorption, muscle wasting diseases, severe burns caused by loss, renal disease caused by loss, starvation, and malnutrition. Increased in dehydration. Alpha-1 antitrypsin is an acute phase reactant and a protease inhibitor that neutralizes trypsin-type enzymes that can damage structural proteins. Decreased in emphysema-associated pulmonary disease and severe juvenile hepatic disorders that may result in cirrhosis. Increased in inflammatory disorders. Alpha-fetoprotein is synthesized during gestation in the yolk sac and liver of the fetus, peaking at 13 weeks and declining at 34 weeks. Normally, adult levels are very low. Maternal serum alpha-fetoprotein is measured between 15 and 20 weeks of gestation and is reported as multiples of the median. Increased alpha-fetoprotein level in maternal serum, neural tube defects, spina bifida, and fetal distress. Decreased alpha-fetoprotein level in maternal serum, Down syndrome, trisomy 18. In adults, increased levels of alpha-fetoprotein can be indicative of hepatocellular carcinoma and gonadal tumors. Alpha-1 acid glycoprotein, also known as orosomucoid, is an acute phase reactant that binds to basic drugs. Increased in inflammatory disorders such as rheumatoid arthritis, pneumonia, and conditions associated with cell proliferation. Decreased in nephrotic syndrome. Haptoglobin is an alpha-2 globulin that binds free hemoglobin and is an acute phase reactant. Increased in inflammatory conditions, burns, trauma. Decreased in intravascular hemolysis because of formation of a haptoglobin-hemoglobin complex for removal by the liver. Ceruloplasmin is an acute phase reactant that is an alpha-2 globulin, copper-containing protein with enzymatic activity. Approximately 90% of serum copper is bound in ceruloplasmin. Increased in pregnancy, inflammatory disorders, malignancies, and with intake of oral estrogen and oral contraceptives. Decreased in Wilson disease, malnutrition, malabsorption, severe liver disease. Alpha-2 macroglobulin, proteolytic enzyme inhibitor that inhibits thrombin, trypsin, and pepsin. Increased in nephrotic syndrome, contraceptive use, pregnancy, estrogen therapy. Transferrin is a beta-globulin that transports iron. Decreased slightly in acute inflammatory disorders and prostatic cancer, decreased markedly in acute pancreatitis. Decreased in infections, liver disease, and nephrotic syndrome. Increased in iron deficiency anemia and pregnancy. C-reactive protein is a beta-globulin that is an acute phase reactant. Increased in tissue necrosis, rheumatic fever, infections, myocardial infarction, rheumatoid arthritis, and gout.